Hello. So I'm going to talk about absorption, distribution, metabolism, and excretion data that's available on the dashboard. That is basically a synonym for what is called pharmacokinetics or toxicokinetics, and that allows translation between the sort of bioactivity and hazard data that Dr. Paul Friedman was just discussing and some of the exposure data of what we actually get exposed to that will be discussed later in this series. So I'm just going to go right back to bisphenol A, not out of any particular interest in the compound, just because it's a particularly well-studied compound. So the tab I'm describing to you is labeled ADME, and again, that's for absorption, distribution, metabolism, and excretion. And when we click on it, we get a single page. So my task is a little bit easier than my colleagues here in explaining things. So what we have for bisphenol A, it's the ADME tab over here, and then there's one sub-tab labeled IVIVE. And so again, this is an acronym, and this is for in vitro to in vivo extrapolation. And there are actually two types of in vitro to in vivo extrapolation that are used on the dashboard in the information that's provided here. The first is that we do not have in vivo ADME data provided in the dashboard. We are providing data from in vitro toxicokinetic measures. So that means that we have to do in vitro to in vivo extrapolation to understand the toxicokinetics. We do that because we want to also extrapolate the in vitro bioactivity data, these points of departure in some cases, to in vivo situations. So that's a second extrapolation, in that case from in vitro bioactivity data to whole animal or whole human doses. Everything that we provide right now on this table is actually from is actually for a human. So just want to run down what quantities we have here. Again, you can download this table for an individual, and you can get individual columns displayed or not displayed. The units for each column or for each row are provided, well, actually in a column labeled units, and a greater explanation of each row is provided as you scroll through it. So what is provided here? First, two in vitro measures. So these actually come from in vitro experiments. The first is a measure of human metabolism rate, and so that is using a pool of suspended human hepatocytes. It's conducted over 2 hours, and it is an assay that is often used by pharmaceutical industry to calculate the rough half-life of a compound in the body. So how long is that compound going to stay in you? And the trick with this is that these measurements require some, they require development of chemical-specific analytical methods, like the ones that was shown for bisphenol A from the Tox 21 project. That takes time to develop those methods, and so we add chemicals at a rate of about 100 per year. And so over time, you will see more and more measured values here. If I go to an arbitrary chemical out of the 800,000 in the dashboard, we have not yet measured that. Doing it at 800, or doing 100 a year, we've got a few years of work ahead of us. The second measured value that you'll see is the fraction unbound of that chemical in human plasma. That's a steady state quantity, and again, with these two values, a pharmaceutical company might be able to predict what the half-life of a chemical is in your body, how much of a dose you would need to be efficacious. We then have a column that for bisphenol A, it's not currently populated, it's predicted values. There are a number, because of the gap in the speed that it takes to measure these values, there are QSARs, quantitative structure-activity relationships, or in this case more properly QSPRs, but that's harder to say, that will predict these properties. Where those predictions are available, and right now the main publication that we draw from is a CITES 2017 Environmental Science and Technology article. We will include the predictions of these values, and then with those predicted values, we can make computations of key properties of interest. So the first is volume of distribution. That is a term of trade in the pharmaceutical and toxicokinetics industry where it allows you to relate the serum concentration that you see of a chemical to the total body burden of that chemical. The next is the days to steady state. So this is how long you would need of a constant dose, or not a constant dose, a frequent dose of the compound to achieve steady state. In this case, for bisphenol A, you would achieve steady state after 8 days. It's a relatively rapidly cleared compound. We then have a predicted chemical half-life. So the chemical half-life depends on this volume of distribution and these measured clearances. And so the chemical half-life is measured in hours, and it's basically how long does it take, if I get 1 pound of the chemical in me, how long does it take for there to be half a pound of that chemical? And so this is a prediction that's based both on the measured values and then the predicted volume of distribution. The volume of distribution is actually predicted using QSA, or using chemical informatics techniques to try to predict how the chemical partitions into different tissues. Does it partition into lipid? Does it partition into muscle? Does it partition into nothing? The paper that describes that method is, Robert Pierce is the first author, and appeared in 2017. The final is the most important quantity. The final quantity given is the most important quantity given on this table, and that is the human steady-state plasma concentration resulting from a constant infusion 1 milligram per kilogram per day dose. That seems like an awfully tangential thing to be the most relevant thing, but that's actually what you need to know to do in vitro and vivo extrapolation, is that quantity at the bottom. So here we have a number of 1.98 milligrams per liter. So if you wanted to use this value, you first need to convert it into a micromolar value. That involves just dividing by the molecular weight. You then can take this value, and this is the concentration I get from a known exposure. Well, if I want to take that cytotoxic 10 micromolar that was putatively identified in the previous presentation and convert that to a human dose, I take 10 micromolar and I divide by this quantity, this steady state concentration. That gives me a dose in milligrams per kilogram per day that would produce 10 micromolar at steady state. And then your in vitro and vivo extrapolation, your very efficient high throughput version, is done. You now know this is a dose that causes 10 micromolar. And so that's why I say this is the most important quantity on this in vitro and vivo extrapolation page, is that it's trying to give you that value. Now, one thing I will point out, if you want to know what chemicals are available, to do this, you can certainly go one chemical at a time through the dashboard, but there are lists available. The general term we use for this is HTTK, that is for high throughput toxicokinetics. And so there are 2 lists on the dashboard right now, one corresponding to a paper by Barbara Wetmore that appeared in Toxicological Sciences in 2012. That gives you a list of all the chemicals that were in that paper. So 238 were measured. There was a 2015 paper, another 178 were added. There's a 2019 paper that I happen to be the first author on, so Wambaugh 2019. We are in the process of adding that, and you'll see that adds another couple of hundred chemicals. So this is where you see the chemicals being added over time. You can see the lists, or again, you can go to the dashboard and click the Add Me tab and find the chemicals that you need.